I'm Ameen Eetemadi. I come from University of California, Davis. I work with Dr. Ilyas Tagapoulos. Today I'm going to talk about self-learning agents. Now, self-learning agents are agents that do not only sit for you to give them data, and they would just use whatever data you give them, which is many of the machine learning techniques are designed like that. Self-learning ones are the ones that will know what data they want. What is the data set or what is the data records that they need in order to expand their knowledge. So they are, in that sense, they are making an effort to accelerate their knowledge discovery. But before I go into some of the details, I would like to say what are the things that I use Blue Water for. Generally use it for two projects. The first project is to design artificial neural networks that can capture gene expression regulation. This is in contrast to many neural networks which you do not necessarily, it's not easy to understand what's going on inside them and how you relate them to the actual application. Here you not only understand what's going on, you can also get much better predictive power. And secondly, is closely related to the self-learning agents, which is to determine what is the best strategy to identify the next set of experiments you need to do in order to accelerate your knowledge discovery. And specifically, I run thermodynamic simulations on Blue Waters. I run deep learning and many evaluations, including parameter sweeps and validating with respect to different data sets and different competing algorithms. And similarly for the second project, but also I use RNA-seq data processing, synthetic data generation, and so on. Now that we talk about these self-learning agents, what is it that they try to accomplish? They try to discover knowledge. Now, I think we live in this era. There are four important technologies that come together that will make knowledge discovery much more accelerated than before if we take advantage of them and put them together nicely. We can get benefits that we've not seen before. First is the high-throughput. First is the high-throughput omics. That is, you get very detailed information about the cell. Is transcription, proteomics, more than ever before. So you get a lot of data, and very high definition data. Now, can we directly process them? Can we directly understand fragmented read? We cannot. We need to rely on the algorithms. In this case, we rely on artificial intelligence to help us extract knowledge from this data. Now, the advances in robotics, it allows us to perform experiments faster, make them more efficient, make them more accurate, and the fourth one is the high-performance computing, which enables analysis of the high-throughput omics data and also running machine learning algorithms, which can be very computationally extensive. So putting all these together is going to accelerate our knowledge discovery. Now, specifically in our project, our goal is to not only do knowledge discovery, but make sure it's efficient. That is, we want to maximize our knowledge about, in this case, the bacterium E. coli. You can do it with other species, but we currently focus on bacterium E. coli as a first model. At the same time, we want to make sure the cost is minimized. Now, you know there are many applications just to bacteria itself. Set aside just this idea of self-learning, but for the bacteria E. coli itself, it has many applications in medicine, relates to diseases, relates to drug production, such as insulin. It's important for food safety. You once in a while hear about the E. coli outbreaks. And also for basic science, where this bacteria is many times preferred for many of the experimentations in various labs. Now, to do knowledge discovery, we need to understand what are the basic components in the knowledge discovery when we want to discover knowledge. There are three main components. First component is the data. Without data, we don't get any, we don't know nothing. The other part is the lab. If we don't have a lab, we cannot make data. We need to make the experiments. And the third one, which is sitting in between, is how we make sense of this data. So first step is that we learn, assuming we have some data, when we generally have, in any area you go, there is some data. And the intelligent agents will make a model from this data, in this case for bacteria E. coli. Okay, after it made this model, it needs to figure out what are the gaps within its knowledge. That it has acquired some knowledge, it can do some predictions, and what are the gaps within its knowledge. Therefore, it's going to find those gaps and order new experiments, and these experiments that it orders, once it comes out of the lab, is going to be added to as an observed data to the data set, and will continue the cycle until some of the constraints are met. Perhaps budget, perhaps we already know as much as we need to know about the bacteria we are looking at. So the first project was about just the learning part. How can we maximize our learning? Now, specifically in this project, I want to learn the dynamic program of a cell. Okay, that's a very big goal. This is very hard. So we be a little more conservative, and we say we want to learn the dynamics of gene expression, which is one of the facets of the dynamics in the cell. So we developed a algorithm we called genetic neural network, and the basic idea is that we have a genetic neural network. We have a gene regulatory network. In this case, for illustration purposes, we have four genes. A, B, C, D, each are genes, and gene A may be perturbed from some outside signal which affects its expression, and expression of A is only going to affect expression of B directly, and the rest indirectly. Now our question is, what is the expression of the rest of the genes given this network? So our genetic neural network architecture, in contrast to other architectures you may be familiar with, multi-layer perceptron, recurrent neural network, all of which do not necessarily correlate with some existing knowledge about data. In this case, we incorporate this gene regulatory relationships into the network. So for example, the first thing is Well, the first signal is coming from the master regulator, which is the first thing in the network, is going to be in the input. The second thing in the input is going to be whether a gene is going to be knocked out, whether the gene is going to be present or not. And for each gene, the related signal is going to be connected with the corresponding gene. So the middle one, for example, is saying gene C is knocked out, so that layer is informed that it is knocked out. Secondly, for example, for B, you see that expression of B in here impacts 2 genes, and here you also see that it's an input to gene C and also gene D. But for gene B, it's only impacted by gene A. So this information is encoded in the network in the following fashion. And furthermore, these are the activation functions that are used, which I'm not going to go to the math to cover, but briefly, we have 2 key features that I would like to mention. First is our network captures the regulatory relationships. This artificial neural network is able to capture relationships. And secondly, it can model the transcription factor dynamics given the nonlinear function that we used to be able to do this. And we used several strategies, including layer-wise training to make this possible. We recently published this in the Bioinformatics Journal. Now, I'd like to emphasize just some of the results that we got. The first one relates to the chemotaxis transcription regulatory network. So chemotaxis is the part of the bacteria that tells it which direction it should swim using these arms that it has, the flagella arms, which direction it needs to go to find food. So it's very important and very well studied part of the bacteria. And you see the genes, the circles that are bigger here are the master regulator, are the ones that are perturbed through outside. And our goal is to predict the rest of the genes, the expression of the rest of the genes. Now here you see that the GNN is genetic neural network that we designed, and LinGNN is an alternative which is only using a linear activation function. So already you see that the error, the mean absolute error of the prediction is lower than the competing methods. Another interesting factor is that after data sets with size 30 or more, you see no improvement. Meaning that, and we try to make sure each data set is as diverse as possible. But when you add more data, you get more benefit. So this is an important analysis that tells you when to stop. When do you not need? Sometimes you don't need to gather more data. This is another result, which is for real data that we used, and it encompasses various network sizes from 10 genes to 1,000 genes. And similarly, the network that we designed, we're able to get better performance compared to the competing common neural network models. Okay, so we covered one method, introduced to how it could learn from data. Now, want to see the big picture, like everything together in another example of our project. In this example, let me first introduce you, as we talked about experiment, what is the experiment that we do? The experiment is the following. You have bacteria E. coli. You first expose it to some biocides. For example, you expose it to ethanol is a biocide. And after some time, for example, after 1 hour, then you expose it to an antibiotic. Now the question is whether exposing to ethanol initially, whether it is going to build resistance for it or not. And here we see that in some cases it does build resistance. So briefly, the methodology works like this, that we have some data initially. We start with some data, and some are empty, which we don't know anything about. We don't know the gene expressions. In each iteration, we build a predictive model. Then we calculate utility scores, which is finding the gaps in the knowledge. And the parts of that are we are most interested in, and that we don't know much about, and we consider them as gaps in the knowledge. We select them for experimentation. So in each step, we add one data point and move forward in this strategy. So our results show that in each iteration, basically as each iteration you are comparing against the data points that you never seen. You train on the green ones and you test on the white ones, which you have never seen. So for example, at iteration 15, you see that the green, which is the SMART method, which we call the optimal experimental design, which we use here, is achieving lower error compared to the rest of the models. But more interestingly, other sampling methods, including random and the one that designed by an expert without using the gene expression data, they took about 13 iterations, which is close to half of the data, half of the number of iterations remaining. So it takes so much for the other methods to achieve the same accuracy that this method achieved at iteration 15, which is accelerated knowledge discovery by this method and delayed knowledge discovery by the other methods. So we talked about these four pillars that are important for knowledge discovery. And we use Blue Waters for high-performance computing and RNA-seq in terms of high-throughput omics. And we devised one novel method, and we leverage optimal experimental design for accelerating knowledge discovery. Now, before I end, I would like to just summarize the Blue Water experience. One point is our workload was extremely parallel and independent small jobs. And Blue Waters for us was very reliable and highly available and very good documentation. And we thanks to the Blue Waters customer support, which help us with their fast and high quality response. I'd like to thank my advisor first, who initiated the projects and advised us through the project, and also my colleagues Xiaokang, Navneet, and Beatrice, who we worked on these projects together, and the funding that we get from NSF. Thank you very much.